Hello and welcome to this BioProcess International Ask the Expert webcast. I'm your host, Leah Rosen, the online editor for BioProcess International. Before we get started, just a couple of notes. This webcast is being recorded and will be made available for replay in the multimedia section of our website. We've muted the audio lines, but we welcome you to type in your questions for our speaker in the chat window on your screen. After the presentation, we will begin the question and answer portion, and I will ask our speaker your questions from the chat window. Your questions in the chat window will only be visible to myself and our speaker. So thank you for joining us today. It is now my pleasure to introduce our speaker, Antje Schickert from Sartorius, Stedim, North America.
Thank you so much for the introduction and welcome everyone to today's webinar. I will be speaking about the impact of rapid virus quantification in the development and manufacture of emerging therapies. The use of viruses in modern therapies is expanding to a state where virus manufacturing can be a very critical bottleneck. The increasing demand in virus products requires more efficient manufacturing processes to keep pace with the required amount of viruses. This targeted optimization of manufacturing processes requires us to get a very detailed understanding in the virus titers and quality during upstream and downstream production steps. Traditional quantification methods for viruses often cannot keep pace with the requirements of modern cell and gene therapies. The results of traditional methods are often not available for days or weeks, which can slow down or even pause our manufacturing processes, delaying decision-making, and thereby increasing cost. Many of these traditional assays are also inherently variable, which can decrease the confidence that we have in the results they deliver us. Virus samples can actually be highly heterogeneous mixes of infectious particles, empty particles, and defective particles. Samples can additionally contain unassociated proteins or nucleic acid fragments that derive from unassembled viruses. Here we show at the bottom here with the AAV2 reference material from ATTC how different quantification methods can quantify the same sample. This result emphasizes the importance of understanding which sample component each quantification method is actually looking at. Here we use the virus counter technology here in orange to enumerate total virus particle counts, while TCID50 and transducing units are methods that look at infective particles only. That is why these always titer lower than our total particle count. Qpcr and Elisa here on the right side measure virus components such as nucleic acid or viral proteins and derive the virus concentrations from these building blocks. Their results may be biased as they are derived values and they are not based upon direct detection of intact viruses. Therefore, the heterogeneity of the sample as well as the nature of these different quantification assays are responsible for these diverse results we obtain when we quantify one sample with different quantification methods. Infectious particles can actually be a very small fraction of total virus count. Depending on the virus and also the manufacturing process used, the ratio of total particles to infectious particles can be between 1 to 2 to up to 1 in 10,000. You can see here on the left side also the quantification of 4 different viruses with 2 methods that look at total particle counts, the virus counter and electron microscopy. And these are in comparison to infectious particles that are measured with TCID50. And you can see that there is a huge difference between those 2 metrics, and these are the non-infective particles inside the sample. In applications where viruses are directly administered to patients, for example, in gene therapies, it is absolutely vital to characterize these virus samples in detail to ensure the safety of the patient. Quantification of infectious particles alone can actually lead to a vast underestimation of particles administered to a patient. So a good understanding of the composition of these samples can really help to make modern viral therapies safer for our patients. So let me introduce you to the virus counter platform. I already showed you some results on the previous slide. This system is specifically designed to rapidly enumerate total virus particles using both biologically relevant and biologically specific reagent technologies. The virus counter platform includes the instrument, software, as well as reagents, and is purpose-built to enumerate viruses rapidly. Multiple different quantification reagents are available that address diverse quantification needs. System operation and data analysis are software-assisted, which limits the risk of any user error or variability. The virus counter offers a very simple and rapid workflow. As you can see here, the virus samples are directly labeled with a reagent, then incubated for 30 minutes and then ready to be analyzed on the virus counter instrument. There is a more universal quantification reagent available that has very broad utility and can be used with a large variety of viruses. But there's also an antibody-based reagent line that allows virus detection with very high affinity and specificity. Inside the instrument, the stained virus sample here at the bottom of the slide is guided into a fluidic stream and is hit by laser light. This excites the fluorophores that are directly associated with the virus. The instrument then detects the light that gets emitted by the fluorophores and uses it together with the sample flow rate to calculate total particle concentration very precisely. Rapid, precise, and biologically relevant elimination of viruses is critical across several applications where viruses are used as biological tools in support of human health. The virus counter platform enables our customers to get immediate insights into their titers during process development as well as manufacturing of viral vectors. This information allows the optimization of growth conditions, tracking of virus titers in the bioreactor, as well as during downstream purification steps up to the final product. This means the customer has very actionable data that allows the optimization of growth conditions, processing steps, and even the identification of any challenges or problems very early on in the process. In the past, the availability of a quick and reliable quantification method often failed to provide these crucial insights effectively. In a very recent collaboration with Qpcr in France, we were able to show how the virus counter technology can be used to monitor virus titers during purification steps in an adherent and a suspension cell process. We used the virus counter platform here in conjunction with our Biotech VSVG reagent to quantify lentiviral vectors. This Biotech is an antibody-based reagent that detects VSVG pseudotype lentiviruses with very high specificity. Qpcr measured lentiviral vector titers throughout the downstream purification for an adherent cell process. Here on the x-axis, you can see the different processing steps they were titering, and on the y-axis, you can see the virus titers that was obtained. Virus counter results here in orange were compared to ELISA results here in black, and as you can see, they track very well, with ELISA titers being about 10 times higher than virus counter results. And this could be caused by the different matrices that these two quantification methods are looking at. It's possible that there is free P24 protein in the sample that gets picked up by ELISA assays and therefore increase those counts. You can also see the infectious particles here in the light gray. They also track very well with both matrices, but as expected, they are approximately 100-fold below the ELISA results and about 10-fold below the virus counter results. Very similarly, Qpcr ran the same assay here for their suspension cell process, and the results still track closely with each other. The ELISA assay and the virus counter results, they are much more similar here than as seen for the adherent process. The different processes might be impacting the virus sample composition and possibly reducing the occurrence of free P24 proteins, causing the two assays to titer much more similarly. The observed functional titers, here again in a light gray, for this process are about 1,000-fold below the physical titers. And this could indicate that this particular suspension cell process used here is generating a lower ratio of infectious lentivirus particles as compared to total particle count. The collaboration between Qpcr and Sartorius was able to show that the virus counter technology provides a very reliable method for lentiviral vector particle quantification, which delivers results extremely fast, in less than an hour for the virus counter versus 8 hours for ELISA, and is much more precise than alternative methods, while delivering very comparable quantification results in near real time. That means you don't have to wait for other quantification results that could take up hours or days. The virus counter platform enables us to measure virus samples in near real time and with very high precision. This makes those virus counters results very actionable. The technology empowers users to increase the viral vector yield by comparing different growth conditions and the recovery during their process development. Users can track virus titers throughout the whole process. They can determine ideal conditions and even detect any kind of problems very early on. Additionally, the virus counter increases the safety and the efficiency by facilitating an in-depth characterization of total particle concentration in a final product. If you're interested in learning more about the virus counter technology, please visit our website or contact me directly. Our application note, in collaboration with Qpcr, will also be available on our website very shortly. And I want to thank you all for your attention, and I am open for questions.
Okay, great. Thanks, Antje. So the first question is, what is the advantage of the virus counter over the ELISA assay?
With the virus counter assay, you look at total particle quantification. So if you go back to a slide here in the beginning, virus samples can be extremely heterogeneous mixes. So it is important to know what your total particle counts are. And so with the virus counter quantification method, you're looking at infectious particles, empty particles, as well as defective particles. When you're using an ELISA assay, you quantify the total amount of viral protein within the sample. And so this could also be unassociated proteins, for example. And then from using this method, you are calculating from a protein concentration how many viruses you have in a sample. So it's a derived value instead of a direct detection of total particles.
And is the Virustag method regulatory approved?
No, it's a research use only tool at this point.
And what is the sensitivity of this method for virus particles per milliliter? So
our instrument quantification limit is about 5 times 10 to the 5th particles per milliliter. And that has just statistical reasons because we want to guarantee for a very quick enumeration method. So our actual sample acquisition time is only a minute, and so due to statistical reasons, this is currently our limit.
Are there reagents available for different AAV serotypes?
Yes, so there's different antibody-based reagents available, and AAV is one of them. If you're interested in our portfolio, please look at our website or write me a message, depending on, and let me know what kind of viruses you're interested in.
Okay. And can you use the instrument to quantify HPV particles?
So human papillomaviruses, we cannot currently see. We are working on, we are always increasing our portfolio, but HPV is something that is not currently possible.
Are there any issues known with having serum present in the harvest samples?
So with the antibodies have no issue with serum in the sample. We have a more broad reagent called a combo dye reagent that works very, very good with more purified samples. If you would be interested in using combo dye, we would look at serum concentration, but it has no impact on the performance of Biotech reagents.
Does this method quantify damaged capsids?
So with damaged, I'm not sure, do you mean, what exactly you mean? So we currently cannot see the difference between an empty and a full capsid. So the presence of nucleic acid does not currently or you cannot currently distinguish between a capsid that has nucleic acid in it or that does not.
Okay. What is the advantage of using this technique over Qpcr?
Well, it's very similar than the ELISA assay. Qpcr looks at total nucleic acid in the sample. So it's an indirect method from which you measure total nucleic acid in the sample and then derive a total viral concentration. If you have free nucleic acid, for example, in the sample, you would have an overestimation of your titer at the end. So with the virus count technology, you're looking at total particles directly instead of having a derived value from only one building block of the virus.
Can it be used for retro and other adventitious viruses?
So if you talk about adventitious, you might. So we are currently using it for viruses as a product, and the reason is that our instrument quantification limit is at 5 times 10 to the 5th viruses per milliliter. So if your concentration of the adventitious virus is below this, we might not be able to pick it up. So I would not recommend it for this use at this point.
How about for lentiviruses?
Absolutely. So the study that we performed here with Qpcr was done with lentiviruses. And we have this Virustag reagent called VSVG that works very well with lentiviruses.
Okay. So I think we have time for just one more question. If you ask the question and we haven't gotten to it, Antje will be given all the questions and your contact information, and she can follow up directly. So the last question is, can you confirm when running a sample that all viral particles have been labeled, and if so, with the same amount of fluorophore?
Yeah, so we add more fluorophore, of course, so the sample is saturated and completely stained. We optimize obviously all our reagents in our development and ensure that the concentration of the reagent is high enough to label every single virus and epitope in the sample.
Okay, well, thank you, Antje.
Thank you very much.
And thanks to our audience for joining us. The recorded version of this webcast will be made available for on-demand viewing on our website, and as a registered attendee, you will receive a follow-up email providing you with a direct link. We look forward to having you join us at future BioProcess International Ask the Expert webcasts. Look for those announcements in your inbox. Goodbye.